Turning now to the main feature of tonight's meeting, I introduce our speakers who will tell us about marsupials. Their secrets are all in their genes. As you will hear, this is a subject of great importance to Australia, which bears the risk of extinction of many of our iconic species. Belov and Deane are using genetic techniques to help buffer these species against inbreeding, loss of genetic diversity, and increased susceptibility to disease. Professor Kathy Belov is Professor of Comparative Genomics and Pro-Vice-Chancellor Global Engagement at the University of Sydney. She is a highly published researcher whose work has been recognised by the fellowships I have shown on this slide, Fellow of the Royal Society and the Academy of Science, and by awards that include the Australian Academy of Science Fenner Medal, the Crozier Medal from the Genetic Society of Australasia, and the MJD White Medal from the Genetic Society of Australasia. Kathy's contributions were recently recognised by the award of an Officer of the Order of Australia in recognition of her service as an academic and researcher, especially in comparative genomics. Kathy contributes generously to the work of the Society. She's a member of the Society's Council and Executive Committee and Chair of the Community Engagement Committee. Dr. Elizabeth Deane is a retired academic who now works as an independent higher education consultant to universities and private higher education providers. She spent the final years of her academic career in senior management, most recently as Professor and Director of Learning and Teaching and Dean of University of Western Sydney Online. Before that, Liz was Pro-Vice-Chancellor at ANU. Her research work focused on marsupial immunology and disease, with an emphasis on the development of immunocompetence and neonatal protection. Liz also contributes to the work of the Society as Secretary of our Program Committee. I welcome both Liz and Kathy.
Thanks very much, Susan, and hello, Kathy. I welcome our forthcoming conversation. So Kathy will be doing most of the presentation. As Susan alluded to, I have retired from being an active researcher, so my interest and expertise is somewhat historic. So I will actually be talking us through some of the history, particularly how methods, the increasing sophistication of methods has allowed us to move away from what was fairly immature perspective of marsupial immune function in particular. So I would also like to start this talk by acknowledging the traditional custodians of the land on which we all reside and pay my respects to their leaders, past, present, and to come. And you will see in my opening slide, I've presented a drawing from a cave in the Kimberleys. So obviously marsupials have been of amazing importance to Aboriginal people, both cultural, social, spiritual, and a particular source of food and dreaming stories. So we do acknowledge that it's not just, us researchers that are actually heavily into marsupials. There's a very strong tradition of engagement with our Aboriginal people. I put a nice picture of drawings from the first explorers or first white explorers into this country. So you can see that they were also a source of great wonder. Marsupials, they looked quite different, they moved quite differently, and there are many pictures of marsupials, some made to look a bit like dogs found in Europe, but certainly there are many different species of marsupials that were drawn by the first white colonists. So this talk really is looking at moving along. Good, right. So the position I'm taking in this introduction is that there have been three main drivers to look at research into marsupials. Firstly, I've already mentioned they're very diverse and very different from what was seen in Europe by Europeans in their home countries. There are about 300 or so species, and I'm sure that's a debatable number, found in South America, PNG and Australia having the majority of them. I won't go into the geology, but it was essentially because the continents were stuck together when they arose, and when they moved apart within Australia, a large number were isolated. There were a few placental mammals at the time, but they died out, and marsupials became the dominant types of marsupials, so of mammals in Australia. So they're diverse and they're different, and therefore they became an exciting focus of research by Australian scientists. The second driver for research is their really unique development. You've probably all seen these sorts of pictures of very young neonatal marsupials. They're born very, very tiny, about the size of a jelly bean, with very few mature tissues. Their forelimbs are certainly well developed, but the rest of them, and their sucking apparatus, but the rest of them are fairly immature, and of particular interest for the purposes of this talk have been the immaturity of their immune tissues, and therefore the question of how they actually survive in an environment which is not exactly sterile. So there's been a lot of focus on survival of the young during their period of development in the pouch. And the third driver of research has certainly been disease. And this is where Kathy's got loads of stories to tell, I'm sure. But the ones that everybody would have heard about are chlamydia, which has threatened koalas, the facial tumour in Tasmanian devils, toxoplasmosis in kangaroos, you probably haven't heard that much about, but it is quite fatal, and mange in wombats. They're probably the headliner diseases, but there are many more. But the argument is those three, particularly development disease, have been strong drivers of research in marsupials. Good. Right, the final slide before I let Kathy take over is the basic premise that it has been the advent of sophisticated methodologies and tools that allowed us to move from what was a fairly naive, I suppose, interpretation or understanding of how marsupials' immune tissues worked. So in the 70s, around the 70s, there was a lot of research on marsupials, certainly looking at the anatomy of the lymphoid tissues, that's the tissues of the immune system, and probably the definitive characteristic explored there was the presence of two thymuses, one in the cervix and one in the thoracic cavity. And there were certainly arguments about why two, because some have two and some only have one. It was supposed to be or thought to be linked to the various diets and to protection in the pouch. I don't think that's ever been resolved, but there was certainly a lot of energy put into looking at the reasons for cervical and thoracic thymuses in different types of marsupials. Also at this time in the 70s, standard histological studies, quite simple staining techniques of looking at the development of the lymphoid tissues, the immune tissues in marsupials were carried out. And then some limited functional studies. Well, actually, there were quite a lot of functional studies, but they were often focused on transplants, so tissues taken from one animal to the other, and a lot of them undertaken in the Quokka in Western Australia, where tissue was transplanted and looked at the time to rejection, and essentially finding that if tissue was transplanted early on in neonatal development, the tissue wasn't rejected. So there were some arguments about the maturity of the immune response during that early time. There were also studies of antibodies taken, put into the young to see if they would cross the gut. And there were arguments that early on the antibodies moved across, but this is the time the young came to emerge from the pouch, that transmission ceased. So there were a lot of fairly fundamental studies undertaken in the 60s and the 70s, and they concluded basically that the immune responsiveness of marsupials, both adult and young, were immature compared to placental mammals like us. If we move later into towards the 80s and 90s, More sophisticated histological tools, antibodies, which gave specific reactions to cell markers, were developed, and also cellular studies where cells isolated from the blood or tissues of marsupials could be tested for their reactivity. So those sorts of studies actually led to a conclusion that maybe things weren't as different as thought, but there was still this aura around marsupials that they were immature and lacked the repertoire of placental mammals like us. And finally, we move into the late 1990s, into the 2000s beyond, where Kathy's going to take off, where we find the increasing use of DNA methodology and the advent of proteomics and lots of other omics. But I certainly, before I departed being an active researcher, had a bit of to do with genomics and proteomics, and certainly was a lot more labour in those days. So to isolate 1 or 2 genes could be somebody's whole PhD. It's a lot quicker now. So what I'm trying to show here is that we've moved quite a long way in the last 40 or so years, because the tools have improved so much to allow us to actually compare down at the genomic level and down at the other aspects of functioning. It's given us a clearer handle of how marsupial immune systems function, how they develop, and how they're involved in disease and protection. And so now I'll hand you over to Kathy. Thank you.
Well, hi, everybody. It's lovely to be with you today. I would also like to acknowledge that I'm on lands of the Wallamedical people, and I acknowledge elders past, present, and emerging. And as Liz was talking, I actually worked out that Liz and I actually started working together 27 years ago. Liz was doing a sabbatical in Des Cooper's lab at Macquarie University, and I was a new honours student at the time. So it's really lovely to connect now all these years later and to be able to continue the story that Liz and Des and many others started. So, as Liz mentioned, my passion since those days has been understanding the immune systems of marsupials and monotremes. And I've been very fortunate during that period that the technologies have improved and allowed us to make massive advances in our understanding of the immune systems of our native species. And as Liz mentioned, when I was a PhD student, the general consensus was that marsupials had primitive immune systems and they weren't quite as advanced as placental or eutherian mammals. Now, I struggled with that concept a lot because they've been around for many tens, even hundreds of millions of years. And on the whole, marsupial populations are pretty robust, with some exceptions that we'll talk about. And I remember Des Cooper saying to me at the time, well, prove them wrong, show that their immune systems are the same as our own. And that's what I set out to do all those years ago. And Liz also mentioned how hard it was. So back then we would construct cDNA libraries, which would be a whole process, and then we'd use cross-species probes. So I'd go to the butcher and get tissue, pig tissue I was using. I found pig probes work particularly well. I'd isolate the gene of interest from the pig, and then I would hope that that gene would hybridize to something in my marsupial library. And it was the sort of thing that you would spend years on and get very excited when you saw potential positives or dots or clones that were your gene of interest. And the reason that it was so challenging was because there's been a long evolutionary time period between the separation of the pig and the possum, and the level of similarity between the gene sequences is quite low. So you'd be hybridizing your probes at very low temperatures and picking up all sorts of background. But I must say those days were the fun days because it was very hands-on. You were in the lab for long hours. And when you did get a black dot, like one of these black dots, it really meant the world. So during my PhD, I described 4 immunoglobulin genes in the brush-tailed possum. And then I went on to the Australian Museum with an ARC fellowship and described some more immune genes in monotremes this time. And, you know, I was just starting to eke away at characterizing immune genes. Yes, they were definitely looking quite like placental immune genes. When the human genome was released. The human genome was released in about 2001. You'll remember it cost about 2.7 billion to sequence this genome, but it opened the floodgates. And I got to be part of the first marsupial and monitoring genome projects. They were big international programs, very heavily at the time funded by the US, with quite minor contributions from Australia. But we got to jump in and get involved in the analysis and the interpretation of the data. So what you'll see here, the top photo is the team that was involved in characterizing the opossum genome. And that was led out of the Broad Institute, a moment of forgetfulness. So that was a great project that Kirsten Lindblad-Toh led. We led a program out of Australia, the Tamil Wallaby Genome Project. Many of you will know Jenny Graves and Marilyn Renfrey, who were the leaders of that project. Wash U and Wes Warren led the Platypus Genome Project, which was also massive. And these were in the days where we were still using the old sequencing technologies. So these projects cost hundreds of thousands of dollars and, you know, took a great deal of work and fantastic teams. What was amazing for me at that early stage in my career was we would have 5 a.m. teleconferences every morning. It was pre-Zoom, and I remember lying in bed, you know, contributing to these great programs where we had researchers literally from around the globe contributing different skill sets. And some of you may have seen in 2018, Rebecca Johnson and I led the Koala Genome Project. This project was done entirely in Australia, which was fantastic. We had international collaborators, but we did most of the sequencing here. And it's just been nice to see now that we can drive these projects just as well as anyone else from around the world. And my fascination through all of these genomes has been the immune system and the complexity of the immune system. So I'm interested in how immune gene families evolve, how the immune system evolves, and also looking for and discovering novel immune genes that aren't found in other mammals. And so now, my students are able to sequence genomes on their own entirely in-house and annotate them and characterize hundreds of different immune genes from a single genome within their PhDs. And Warwick, I just wanted to give you a shout out to say we have annotated a wombat genome that was sequenced by others. I don't have a huge amount to say about wombats today, but we have started looking at their immune genes in great detail. Liz also showed photos of marsupial jelly beans or joeys. This has long been a fascination for many of us. How is it that you can do an operation on a joey, pop them back into the pouch, and they never get an infection at the site of the incision? So we've known for a long time that there's something protecting the joeys while they're in the pouch. Now, at this stage, they don't have an immune system. They don't have any immune tissues. So they are getting some protection from mum through the milk. But is there anything else? And this is something that the genomes helped us to understand. So when we started looking at these genomes, we found that we were looking at a range of antimicrobial gene families, but where humans, for instance, have 1 cathelicidin, koalas have 10. So they have gene duplications of these antimicrobial peptide genes that we also have. But each of those genes has a different function and a different specificity. And we've found that these peptides are expressed in the lining of the pouch, in mum's milk, and in the skin of the joeys themselves. And interestingly, depending on the timing of pouch life, or I guess the age of the pouch young, the composition of the microflora of the pouch changes. And we believe that these antimicrobial secretions are very much influencing the composition of bacteria in the pouch. And we've been synthesizing these peptides artificially and testing them against a range of bacteria, gram-positive, gram-negative, fungi, and they're super potent. They all have different activities, but we're finding that some of them, for instance, are able to kill multi-drug resistant golden staph. And we really think that future antibiotics may come from the marsupial pouch. We've been very fortunate to be successful in securing funding through an ARC Centre of Excellence for peptide and protein, for innovations in peptide and protein science, to continue this peptide discovery work. It's not only the cathelicidins, there are also other antimicrobial family genes that are found in pouches. And so what we're doing is we're doing comprehensive genome and transcriptome and proteome projects across a range of Australian animals to discover novel drugs that may end up helping humans or other animals, which is really fantastic. And together with collaborators through the Centre of Excellence, we're going to look beyond marsupials. Glenn King and his team are looking at snakes and spiders, for instance, and we're going to look at a range of native wildlife and identify peptides of interest for drug discovery. There's a bit of a photo of the project team. The other thing that I'm finding really quite interesting at the moment is we've started studying the composition of koala milk. So many of you will know that koalas were recently listed as endangered. That came about, or I guess, you know, that listing followed the bushfires that we saw pre-pandemic now, so it seems a while ago, but the bushfires really devastated our koala populations. And in addition to that, our koala populations are under threat due to urbanisation, roadkill, disease. But we do see a lot of koala joeys coming into care, and a lot of those joeys succumb quite unexpectedly to infections. And we know that getting the composition of the milk right is going to be really important. So we've done a lot of work studying immune genes at different time points during lactation, and we've characterized 850 different immune genes that are switched on and off at different time points. And we really want to get to the point, as part of this Centre of Excellence and together with the Koala Hospital, to be able to provide orphan joeys with lactation stage specific artificial milk. That'll be a real goal for us. Okay, so I talked about characterizing genes. After characterizing those genes, we moved quite quickly onto studying diversity within these genes. And in particular, the relationship between genetic diversity and susceptibility or resistance to disease. And pre-pandemic, I used to spend quite a lot of time trying to explain this, but I think now absolutely everybody on the call will know that genetic variation plays a critical role in disease. You know, we all watch to see the latest COVID variants, to see, you know, are they going to be more, are the symptoms going to be more severe, less severe? You know, are the vaccines going to be able to recognize these variants? Well, just like viruses have variants, our immune genes have variants. And you've probably, I won't go through those details, but you've probably all, you know, been one of those people who goes down hard with the flu, where your flatmate seems to, get by, with just a sniffle. That's the variation in your immune genes determining the severity, in many cases, of the response you have to disease. And we know with COVID, for instance, 15% of people who get sick do end up with more severe symptoms than others. Now, part of that's age, part of that's other comorbidities, but there is an underlying genetic component, and there's a lot of work going on around the world to understand that. And some genes that I'm going to talk about a bit later on, MHC genes, toll-like receptor genes, are already being associated with different responses to COVID. So I'm going to give you two case studies today around immune gene diversity and disease. The first one's the Tasmanian devil, and the second one's the koala. So Tassie devils are the world's largest remaining marsupial carnivore. They gained that title after we lost the thylacine, and that was a really sad story because they were really, hunted down and exterminated by humans. The last thylacine died of exposure. It simply got too cold overnight in a zoo in Tasmania. So we don't want the devils to go the same way. They went extinct on the mainland about 3,000 years ago, and since that time they've been found only in Tasmania. And we know in recent history there have been some large population crashes where numbers have dropped to quite low numbers and then built up again. But the species is now listed as endangered, and the reason for that is a disease called devil facial tumour disease. I'm going to show a pretty gory picture next, so if you're having dinner, you might want to look away for a moment. So this is an example of an animal with devil facial tumour disease. It's a disease that's spread by animals when they bite each other, and devils jaw wrestle a lot and tend to bite each other around the face and the jaw. That's why quite often the primary tumours are near the jaw or are on the face. But the tumours metastasize and they do spread throughout the body. So you can get your secondary tumours elsewhere. You can also have primary tumours on other parts of the body if that's where they're bitten. But essentially, animals will tend to die within about 6 months because they're simply not able to feed themselves anymore. But they can die of organ failure via metastases as well. So it's a really, really awful disease. So the disease was first seen up in the northeastern region up here near Mount William back in 1996. It was sighted by a wildlife photographer who took some photographs and reported devils with tumours. Unfortunately, at the time, it wasn't taken particularly seriously because devils are known to get cancers. And unfortunately, by the time it was taken seriously, the disease had spread. So it spread across pretty much all of Tasmania. There are still a couple of disease-free populations in the very west, the northwest of the state. But we've seen local declines of up to 95%. Now, this is a really interesting disease because it's not a bacteria, it's not a virus, it's actually a contagious cancer. And the reason that we know it's a contagious cancer is because of some really important work that Anne-Marie Pierce and Kate Swift did, where they looked at the chromosomes of devil facial tumour disease cells. Now, if you look at the chromosomes here, these are normal devil chromosomes. So you can see there are 6 autosome pairs plus your 2 X chromosomes. So this is a female carrier type. And interestingly, devils have a genome that's a very similar size to our own, but all of that genetic material is condensed onto bigger, chunkier chromosomes. When Anne-Marie and Kate started looking at DFTD cells, they saw a very different pattern, and it looked a bit like this. So what you can see here is both chromosomes 1 are missing, but you can see bits of chromosome 1 in yellow over here on what we call these marker chromosomes. Similarly, you've got little additions here on chromosome 2, additions, you've got a loss of a 5. You know, your sex chromosomes are gone, but you've got bits of X chromosome over here. Now, to have chromosomal rearrangements like this in a cancer is not that unusual. Chromosomal changes happen in cancers often. But what is unusual is that every tumour that Anne-Marie and Kate looked at had exactly this combination of genes or, you know, organisation of chromosomes. And they were able to conclude that this chromosomal transformation didn't occur individually in each individual, rather it happened once, and then that tumour cell spread from animal to animal. And actually molecular genetic work done by Liz Murchison showed that it came from a Schwann cell, a nerve cell in a female devil, and that tumour has passed from animal to animal over the last 20 plus years. And when Anne-Marie and Kate published that paper, that's when I got really interested, because I studied the genetics of immune response, yet that didn't make sense. How can you have cells from one animal passing to another without invoking an immune response? And there is no immune response at the site of the tumour. And so this is what started the journey for me. Now, to understand that, we need to talk a little bit about the MHC. The major histocompatibility complex is a key region of the genome involved in immune response against infectious disease. So it plays a role in, you know, severity of symptoms to various illnesses. But you may have actually heard of it because these are the genes that you're typed at if you need a kidney transplant, if you need a transplant, because you're more likely to get a transplant from a close family member because you're more likely to be more similar at these MHC genes. The more different you are, the more likely your immune system is to kick in and reject the tissue. So these MHC molecules are found on the surface of all cells, and you can imagine here if this was a COVID-infected cell, this cell would just be presenting a bit of virus to T cells of the immune system. Now, the MHC is remarkably complex. You're looking at a couple of hundred genes that have evolved by gene duplication, and we have lineage-specific expansions of genes. So the Tama wallaby genes are very different to the koala genes, are very different to the wombat genes. So you really need to dig into the genome to characterize these genes, to be able to study them in a particular species. And that's what my team's been doing for 20 plus years now. So this photo shows 3 of my first students, Hannah, Claire and Yola, and they together were able to show that MHC diversity is critically low in Tasmanian devils. And you'll remember I mentioned that they'd been through some big population crashes. Well, we think those population crashes resulted in genetic bottlenecks. And because of that, they were very similar genetically, including at their MHC genes. And not only that, because the tumour came from a devil, its MHC type is very similar to the devils it infects. So we concluded at this stage that the devils don't recognise the tumour cells as foreign, and they don't mount an immune response against the tumour because of this similarity in MHC genes. We published that work and we thought, okay, well, to confirm that, we should be able to do some tissue transplants. And Liz mentioned that this was something that, you know, was routinely done. Except that, of course, by the time we were doing this sort of work, nobody was doing tissue transplant experiments anymore. So we had to try to find somebody who was prepared to take some tissue from one animal and graft it to the next. And we did find a plastic surgeon actually in Tasmania who was able to do the work, Frank Kimball, but it was quite a process to work out how to do the transplants. So what you've got here is an autograft. So an autograft simply means it's this bit of tissue that's it's been moved over here. So it's the devil's own tissue, and we're just reattaching it to see that there's no complication or rejection due to the surgery procedure. And then we've got an allograft. So this bit of tissue is a bit of tissue that's come from a genetically unrelated animal. And normally in any other species, if you did this, you would expect this allograft to be rejected very quickly. But we were hypothesizing, because we'd found low MHC diversity, that maybe that rejection wouldn't occur. Oops. We got to 14 days after surgery, and we thought, yes, this is our Nature paper. They're both healing quite nicely. This is looking good. And of course, we drank our champagne too soon, because a couple of days later, the rejection started. So this is a late rejection, but it is rejection. These punches are just holes, biopsies that Alex took to do some immunohistochemistry. But you can see right around the allograft, you've got evidence of rejection of the tissue. And 3 weeks after, you can see it's quite clearly rejected. And I mentioned Alex did some histochemistry. The brown dots show infiltrating lymphocytes. So in the autograft, there aren't many. In the allograft, you've got all these lymphocytes coming in to reject the tissue. This doesn't happen with the tumour. So we knew then that it wasn't simply just that low MHC diversity was at play here. Now, you might remember Hannah's photo. Hannah was my first PhD student, and she went on and continued this work during her postdoc. And she was able to show that the tumours were finding a way to switch off the MHC on their cell surface. It's a bit like Harry Potter and his invisibility cloak. The tumours would just hide from the immune system because they wouldn't be displaying their MHC molecules. And she showed that they were doing this by down-regulating or switching off key genes in the antigen processing pathway. And what's really cool is Hannah was able to show that she could switch this MHC expression back on in DFTD cells using interferon gamma. And so this is actually what we use as the basis for the vaccine that's currently used in Tasmanian devils. MHC is switched back on the cells. That's used in a vaccine, and that in itself triggers a bit of an immune response in devils. Things get weirder though in devils. In 2014, a second transmissible cancer was discovered. What are the odds that another cancer could emerge that is also transmissible? It doesn't seem to have taken off quite like the other one, but it did arise. It was found in the Channel region. And interestingly here, the MHC class 1 molecules were still switched on, but there was some evidence of immunoediting starting to happen. So it's quite interesting because there's something in devils that makes them predisposed to contagious cancers. And there are the other well-known case of a contagious cancer. There are some in mollusks and shellfish and the like, but there's a very well-known contagious cancer in dogs called canine transmissible venereal tumour. It's a sexually transmitted cancer, a cancer cell that's transmitted just like DFTD. It evolved in inbred wolves and also presumably with low MHC diversity. And then just like DFTD, it evolved immune evasion strategies. And CTVT evolved into an amazing parasite because when the tumour is growing, the dog becomes more sexually active. But in healthy animals, sort of within 6 months, the MHC genes kick back on, the dog's immune system gets rid of the tumour and the dog recovers. But by that stage, they've already transmitted the disease. So it started in wolves, but it spread into jackals and coyotes and dogs. And, it's found in dogs on Indigenous camps in Australia. It is out there. Whereas with DFTD, I don't know that we have enough devils out there for this to evolve to a point where it's coexisting, but it's possible. So we've also done some fun stuff like look at when devils lost their MHC diversity, working with Jeremy Austin, and this is my student Katrina Morris. We looked at MHC diversity in fossil samples, in ancient DNA samples from the mainland, and we found some variants, some new variants from 3,000 years ago. So if we have time, we may want to discuss, should we be resurrecting some of this lost genetic diversity? Another big focus for the team has been using this genetic data to manage devil populations. And this work has really been led by Carolyn Hogg from the team. Carolyn's also a fellow of the Royal Society. And I will just go through a couple of examples of the types of things we can do. So very early on in the Save the Devil program, animals were brought in to captivity, mainly on the mainland, but also in wildlife parks in Tasmania. And initially, most of the samples were collected from the northwest. That's because they were disease-free populations at the time. But then animals started getting collected from across the range. And what you can see here is that the pink animals here, the pink dots, are eastern animals, and the blue dots are western animals, and they're quite different. So what Carolyn did, and she really led this project, is we set up an insurance population on Maria Island, which is an island off the east coast of Tasmania, where we mix these eastern and western devils. And you can see the light blue colour are the progeny that were born on Maria Island. And you can see there's much more genetic diversity because we are mixing these populations. So we're boosting genetic diversity in a genetically depauperate species, which is quite exciting. And they're holding that diversity through subsequent generations. This has been a big part of Elspeth's PhD that she's completed now. And Rowena, for her PhD, was looking at what happens to MHC diversity in these populations, because we also saw an increase in MHC diversity in this Maria Island population, and with that, a decrease in parasite load, which is really quite neat. Now, Maria Island, of course, was an insurance population. There's no disease there. There were no devils there before they were introduced as part of this project. We then decided, through work supported by a linkage grant, to move animals from Maria Island into Stony Head, which is a diseased population in Tasmania. And the reason for that is you can see that the Stony Head animals in pink here had very, very low genetic diversity. The blue dots are Maria Island animals. So Carolyn and Elspeth drove this project, and you can see here the light blue dots are animals that were born in Stony Head. The ones in the middle are crosses between Maria Island and Stony Head animals, and that was about 30%. You know, some of them were Stony Head crosses, some were Maria crosses, but about 30% were Stony Head. And we're just looking through all of the immunological fitness data and other data. That'll be a paper that's coming out very soon, but the results look really exciting, because this is a good example of genetic rescue in the wild, where we're improving the immunological fitness of an endangered population. So I'm just going to move on now to koalas. So this is work that I led together with Rebecca Johnson. Rebecca's also a fellow of the Royal Society and now chief scientist at the Smithsonian, which is fantastic. But beyond sequencing the genome, which took many years and a lot of work, we've now started looking at the impact of diversity within these immune genes. And we've found an association between MHC type and mate choice in koalas, where a particular locus had a stronger influence than others in the mate that the animals will select. And we think this will help us improve breeding, particularly in captive and zoo situations. We've also been doing a lot of work on chlamydia. Now, Many of you will know that chlamydia is a real issue for koalas. And sort of like with us with COVID, some people are asymptomatic when they're infected, others have very severe disease. So they can have anything from a mild conjunctivitis to complete blindness and a range of impacts on their urogenital tract and respiratory tract. And we think at least part of that is related to genetics. Part of it will also be stress and environment. So this is Dennis. Dennis actually used a technique called RNA-seq to sequence all of the genes that were switched on in the eye of a koala infected with chlamydia. And he found that 1,500 genes were switched on in a badly infected eye compared to a healthy eye. So there's some information there about the types of genes that are involved. But one of them was interferon gamma, which of course we came across when we were talking about Hannah's experiments. And interferon gamma is an interesting one because you need it to clear the chlamydia infection, but too much of it can cause the scarring that we see in some animals. So this work will help inform future development of vaccines and the like as we better understand the role that these immune genes play in the disease progress. Now this is Luke and Paris. Luke and Paris both did their PhDs looking at the role of MHC in response to chlamydia. And Paris identified genes that played a role in different levels of immune response to a chlamydia vaccine that Peter Timms and team had developed. And Luke has recently, just in the last year or so, published a great paper identifying genes that are involved in both progression and also resolution of chlamydia. What we have done recently, which is also very exciting, is secure funding to sequence 450 koala genomes from across the east coast of Australia. So this is work that Carolyn and I are leading, and it's, we think, the biggest genetic or whole genome sequencing project in a wildlife species to date. It's funded by both the federal and state government, and we're going to use this information to really help us to inform how we manage koala populations across the country. Can we bring in animals, for instance, that are better able to clear chlamydia into populations that don't have that variation to boost the resilience of populations? You know, can we rescue populations that have lost diversity because they've been isolated for extended periods of time? Where you want to go beyond the immune genes and look at genes involved in tolerance of heat, for instance, to boost resilience of animals living in a climate that's getting hotter and hotter. So massive thanks to our partners there with the state and federal government, AWS Illumina and Ramachodi. The genomes are actually sequenced now, and we've made them open access so anyone can jump on and analyse them. They're also very welcome to join our team who will analyse and write a paper, but there's lots of data there to start looking at. So watch that space. Maybe I can come back and talk about that in another year or so. And look, I've made this sound like a very simple story because I was just conscious that I didn't have a lot of time. But I do want to point out that this work has been done over 20 years, and I had a bit of a count before I jumped online, and I think I've had about 70 postdocs and students working on this project. And I just wanted to show some photos. I've supervised some amazing people, and particularly amazing women, and many of them are leading groups themselves. I'm sure you'll recognise some faces on there. And it's just nice to be able to showcase all the great work that they've done and the wonderful way that they've collaborated to achieve that. And finally, I don't have time today to run through all the collaborators and all of the partners and all of the funders. There are too many of them to name, but you can see the logos there. So there are my contact details. And otherwise, I will stop sharing my screen, and I'm very happy to take questions if you have any questions.
Kathy, there are a couple that have popped up on the Q&A, but I might just jump in before you
Fantastic.
Yeah, just want to ask you, we had a great obsession with cytokines when we started sequencing genes, but they seem to have, apart from the interferon here, we seem to have dropped off the radar. I mean, one of the issues was, is there a difference in regulation of the genes? Has that gone anywhere?
So we've characterised the genes, but we haven't really looked at promoters or expression at this stage. So I think it's all there for somebody to jump onto. We got carried away with the diversity side of things, but yeah, I agree. There's a lot to do there.
Okay, well, I have one other, but I'll let somebody else have a go. So can you see the questions, Kathy? All right, I'll read it.
Yes.
So
yeah.
You can see. So the first
Yes.
one is from Ken. Yes, please. Do you want to answer that?
So that's the humble...
Assuming devils have a relatively high breeding rate.
Okay.
Yeah.
I went straight to Warwick's. At least
no, do them in order.
Okay, they're in a different order on yours. Okay.
Oh, are they? Oh, okay.
Yeah, must be. Yep, that's okay. Assuming that devils have a relatively high breeding rate and therefore numbers of generations, reduced rate of random mutations. Yeah, that's a good question, Ken. We don't see much mutation or much variation in the MHC at all in devils. If we look at MHC2, they're pretty much monomorphic. In MHC1, you've got the odd mutation, but not much there at all. So it's really quite unusual to have such low levels of variation and, yeah, lower mutation rates there.
Right, I think Warwick's is just a comment, so it gives me, I think something else popped up. Two questions. How did you first become interested in marsupial or monitoring genomics?
Okay, two questions. Sorry, I was looking at the history of the Professor of Anatomy. That's very cool, Warwick. Thank you. I'm going to ask you about that next time we chat. So how did I become interested? It literally was so I guess Des Cooper had a really profound impact on my career there. I actually did my honours in human genetics on preeclampsia in Des's lab. And after that, he offered me a job and I was just working for him as a research assistant, doing all sorts of population genetics work, very routine consultancy work. But it was while I was in the lab and Liz was in the lab at the same time that people were just talking about primitive marsupials. And I just thought, that doesn't make sense to me. So that got me really hooked because I just can't see how marsupials could be primitive. In many ways, their immune system has evolved in such a way that they're actually more advanced than us. They've actually got these novel protective mechanisms for their joeys. So I think I just got hooked and I actually cloned my first immunoglobulin gene before I even enrolled in a PhD. And once I got a taste of it, I thought, hey, I could make a difference here. There's a huge area to start working on where there's very little information. And it was good timing because just as I came out of my PhD, the genomics technologies came into play. And that was before the disease issues really cropped up in a significant way. So I was well placed once the diseases hit, particularly the public eye, to be able to jump in and study the diseases. So, and is there work going on epigenomics or microbiomes? Epigenomics, no. And funnily enough, Jenny Graves called me today, and that was one of the things she was talking about. Marsupial microbiomes, yes, we're doing a fair bit of work. We're focused both on the gut, and the gut interests us because of the changes to the gut microbiome when we bring animals into captivity. So we've been quite worried about bringing devils in, breeding them up, and are we going to lose that unique microbiome. But we actually find that their microbiomes revert quite quickly when we put them back out into the wild, and we don't see sort of long-term damage or long-term changes in animals that have been bred in captivity. We've also been really interested in pouches because we think that by studying the changes in the pouch during lactation, we can get a better handle on what bacteria our antimicrobial peptides kill. And of course, with these sequencing technologies, as you know, you get so much data that we're just drowning in data at the moment, trying to get our heads around what all these bacterial sequences mean.
Well, as someone who spent a lot of time sitting in koala sheds washing pouches to look at antimicrobial activity, I think the gene route is much more, faster, more advantageous, less labour intensive somewhat.
And cleaner.
Okay, so you mentioned immunological fitness indicators. Would you elaborate on what they are?
Yeah, so we've been looking at everything from lymphocyte counts to body condition to hormone levels to number of joeys born. So we've looked at a range of different measures, microbiome, diversity, cytokine levels. So yes, we've had a team of postdocs generating all sorts of data that's coming together for this Stony Head paper, and I think it'll be a good one.
So harking back to those original transplant experiments in the quokka in the 70s, and they were on pouch young, they were autografts, allografts. Have you ever thought to revisit them, knowing what you know now about looking at, the gene expression that might be going on at that time?
I haven't.
It's all right. I'm not trying to convince cause a new project. You've got enough going on. But
yeah, look, it's an interesting one because I think in many ways we've lost the skills that were around back then. Many of the students coming through now don't do anatomy at all. And yeah, I think it's a shame because I think, we do go through these waves where, all of my students can code and manipulate data, but not many of them would be able to go and, none of them would be able to go and do an allograft. And we found it quite challenging with the devils just to find people who have that skill set. So there's probably only a handful of people in Australia when you think about it now, and many of them are approaching retirement age.
Or past it. So in a similar vein, the story of the cervical and the thoracic thymus, have you thought of looking at using genomics to go back and have a look and see what might be the difference?
It's a good question. So we did, and I had one of my PhD students, Emily Wong, look at differences in expression of genes in the cervical and the thoracic thymus. And no, we didn't see any significant differences. They looked quite similar. That was done, in the mid-2000s, and I think now we've probably got greater sequencing power, so we could probably do it in a larger number of samples at a greater number of stages of development. So it might be an interesting time to start thinking about some of those experiments again.
Yes, though there's the whole lot of experiments around the mitogen responses and all that sort of thing too. So I think
we might need to bring you back into the lab.
No, I don't think I'm very competent in a lab. I'd just kill a few people. So I'm trying to see, we haven't got any questions. So I had one more about this project you've recently embarked upon called the Earth Biogenome and the Three Bridges Initiative. Would you like to tell us about that?
Sure. So the Earth Biogenomes project is a massive project to sequence all of eukaryotic life on Earth. It's led by Easy. But
nothing like a big ambition.
yeah, but the funny thing is
Yeah.
that it's actually going to be cheaper than sequencing the human genome was in the first place. So it's very achievable. It's being led by Harris Lewin out of UC Davis, and I'm on the steering committee. And I think what's exciting about this project is the technology's there. The costs aren't that great anymore. Our biggest challenges are getting the samples. To get access to those samples, to preserve them well enough to get good quality DNA is our biggest challenge. But yeah, you should check out the Earth Biogenome website and just see how quickly entire genomes of many species are being sequenced at the moment.
Make all those poor PhD students who did one gene for their PhD. I'm really jealous.
now you can do a whole genera
in an afternoon.
And so the Threatened Species Initiative is one that Carolyn Hogg is leading, and I'm also involved in, and that's a great one. This is an Australian initiative via platforms, NCRIS funded. And it's really to generate this sort of level of sequencing data as we have for koala and devil for all of our threatened species. And we've got about 80 species that we're working on as part of a consortium. So if people want to get involved, they can just let us know. And we do have funding through this NCRIS scheme to support that work. And our plan is to get to a point where conservation managers themselves can learn how to manipulate and interpret that data. Because I think at the moment that's the biggest challenge, that, there are a limited number of people who know how to interpret the data, but we want to set up websites where you can sort of plug your genome in and get your genetic diversity information out. So that's the plan.
So how does that sort of training feed back into potentially undergraduate or postgraduate coursework programs? So informatics courses. Obviously, there's going to be a need for those sorts of people.
Absolutely. Yeah, huge need. Anyone who has coding skills should definitely get into bioinformatics. There's great careers out there. And, I was showing photos of my students and they're just picking up stellar jobs. So there are great opportunities in the genomic space. And people have moved from the animal genomics into human genomics and the other way around. It's all very transferable.
Good, great. Now we have got one last question, which is, given the sophistication, is there more opportunity of non-coding regions versus the gene region? I presume too.
Yeah. Look, we haven't even looked at the non-coding regions in marsupials because we've just got this massive genetic data to look at. But yes, absolutely. And actually Emily Wong, who's one of my former PhD students, just won a Snow Fellowship to look at the dark matter or the dark genome in across evolution. She's particularly interested from the point of view of heart disease. But yes, I think it's a massive area and we've got the data now. We just need people to jump on and ask the right questions.
Great. Right. Thank you, Kathy. Well, I think we're going to hand over to Christy.
Look, this has been a fantastic discussion and it's been riveting for me because I've been going back to the beginnings of the Royal Society, which of course, was obsessed with the fauna of this new continent, with the marsupials and so on. And we're still leading, but it's a very different space. Peter Tyler wrote about our society, calling it science for gentlemen. Well, look, quite frankly, neither of you two are gentlemen. And it's been really interesting watching the fact that we have two women leaders in this field, in this fast developing field. You are old colleagues. Each of you have been enormously successful, full professors, great research careers, and really senior administrators. Liz has been a Pro-Vice-Chancellor at ANU. You've been acting DVC at Sydney. I mean, these are the top jobs in Australian academia. From this area, which is so quintessentially what the Royal Society is about. So I'm really thrilled to be here. I checked today, and it was only in 1935 that women were accepted into the Royal Society. 1953, before we had a first female president. So, Susan, you're in a good tradition, but a short one. Not that short, since I was born in 1953. I knew Liz when I became Dean of Humanities at Macquarie. And Liz was the most welcoming of Deans of Sciences. And she said, do come over to the science faculty. She said, come to my office, about 5 o'clock when we, sort of 5:30 when we finished work. So I went over, and it was quite hard to find your way, those new universities that no signs or anything. And I finally got to the Dean's office, and there was a fridge, and I thought, oh my God, she's really sophisticated. There's a glass of wine in there. And she said, kangaroo poo. So that's what she had in her fridge. I think what I've learnt tonight is this acceleration of understanding of these marsupials, of their genomics, what we can do with the genomics, how important it is. And I think also, and Kathy, I don't know whether this is fair, but it looks to me that apart from, was it Dennis and someone else, Luke, it's an extraordinarily feminized space.
It is. There are a few other boys there, and we've got 4 at the moment. So we're trying to bring them in, but the women in the team are so intimidating and so amazing that it is a bit of an intimidating atmosphere to work in.
So we have chlamydia, these cancers, we have these extraordinary stories which have, I think, probably gripped everybody as they have gripped me, because they're so important. And I just want to say thank you to you both, because I think it's a story which I don't think I realized before I began was going to be quite such a illumination about what has happened in that last 40 or so years of study in Australia, that we've begun to understand about how important this gender story is, how these stories keep going, and how mentoring is so important. So a riveting story. You're quite right. I gave up my dinner when it got to the picture of your Tasmanian devil. I thought, no, this is not me. But thank you very much indeed. And thanks from the whole society. And I'll hand back to the president to close the meeting.